one of the benefits of synthetic biology, and that's something that we're trying to work very hard in here in Manchester, is how you can provide cells with new functionalities. So we're talking about how you can control things, how you can control behavior of cells in a dynamic way. So you can develop genetic circuits that can help the cells behave better in certain period of the culture, and how you can modulate things in a more controllable way.
Hey, smart biotech scientists. We are in the middle of an exciting conversation about the latest cell factory research. My today's guest is Mauro Torres, who is a chemical engineer and molecular biologist, and his pioneering research at the University of Manchester aims to create innovative technologies that streamline the development and facilitate the scaling up of biopharmaceutical and advanced therapy production processes. If you haven't listened to part 1, please go back. And today we're continuing our journey. And before we do that, what did you think about part 1? Please let us know by leaving a review, and enjoy now part 2. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brolman, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. If we look now inside the cell, what's going on, because there are obviously a lot of different steps involved in the secretory pathway. Can you summarize what's going on there and how these steps influence how much protein finally secreted?
Yeah, as you say, it's exactly when I tried to do the similar of the automobile industry. It gives you some kind of perspective of how complex the manufacturing pipeline could be. If you summarize very quickly, you have different parts of the cells. Like you have a cell that they have a recombinant gene in the nucleus, and that gene is expressed. So those transcripts are translated into proteins, and those proteins are like co-translated into what they call the endoplasmatic reticulum. And I always talk about the endoplasmatic reticulum like the initial part of this pipeline in which the proteins are assembled and then are like early glycosylated and quality control check at the end. And when they are fully formed in terms of, for instance, the heavy and the light chains, you have a specific quality control checkpoint at the end of the endoplasmatic reticulum, and they say, when they check the glycosylation, the specific glycosylation, say, okay, this is ready to move on, or you need to go back and just do some modifications. And when it's ready to go there, it goes to specific vesicles transport into the Golgi. And in the Golgi, you have more refinement. So if you do the similar to car manufacturing, it's like when you go to the point that the car is ready to just mainly assemble what you're talking about, doing the painting, doing the refreshment of the interiors. So you're talking about the refinement of the final product. And when you reach all this refinement process in the Golgi, is you have another quality control point in which the cells say, okay, this is ready to go. And when it's ready to go, it's exported through vesicles to the extracellular environment. It sounds very simple in terms of like, there are a few steps, but there are thousands of proteins, which are the intracellular workers that are working together in a coordinated way to assemble, to check the quality, to add specific attributes to the proteins. So it's a very complex process, and there are so many players involved in the process that sometimes just lacking a few players particularly make completely the difference. And just coming back to the question that you did before is, if you understand what is going on with your cells, you can start designing guided interventions to solve some intracellular problem. And then you can see massive difference on the product titers or in the product quality at the end of the process.
And your research group at the University of Manchester uses a systems level approach, so to gene expression, the host cell physiology. You have done also a lot of omics, all kind of omics. Can you explain in a nutshell, obviously, I know it's very complex, but in a nutshell, how this improves the overall effectiveness of the biopharmaceutical production?
Yeah, I think you got a very interesting point that it is very complex, and it's technology that for some of the omics is very robust. I'm thinking about, for instance, genomics or transcriptomics or metabolomics. It's getting to a point that is extremely robust, and it has become very streamlined in terms of a lot of companies and academic labs have the access to do this kind of analysis. But there are other layers of information in which it's still very difficult to get reliable information. And we're talking about, for instance, proteomics or epigenomics, that I understand omics as like a sandwich in which every omics is like a layer of information that if you put together, it makes sense like a really nice sandwich. Omics data in general is very powerful to understand biological phenomenon, but also could be very misleading because so many reasons. So one of the reasons is the technology is not reliable enough. And coming back to the point, is like how we can use that information for making a better process or better cell factories. It's a very nuanced approach, I would say, and it has to take with a lot of cautious in terms of like how you can really translate that information into real world applications. So we have examples, for instance, of if you have like industrial feedback process and you see that cells, they behave really well until day 10. So your product quality is consistent, you have high titer, but between day 10 and day 14, which is this in the standard way of manufacturing proteins, you have some drop in viability or just variation in product quality, or you start getting less product at the end. A little bit trivial, but like 4 days of GMP manufacturing is highly expensive and highly relevant just to discard and say, okay, why we focus so much on 4 days of manufacturing? But those 4 days, if you keep consistency in the process, it could be very relevant, not only in terms of how you can get the final product, but at the end economically as well. And I guess one of the examples that we have been seeing in our lab, in other labs, is the gene expression of a specific targets that they work in the manufacturing of proteins are very dynamically. So you can have high expression of, for instance, glycosylation enzymes at the beginning, and then some of these enzymes start shutting down at the end, and that results in the impairment of the product at the end. And that's one of the things that we've seen, and you can develop strategies that can compensate that. So you can start feeding specific compounds that they can bypass the kind of metabolisms of a specific glycan products, or you can overexpress or knocking down specific enzymes that they change the expression over the time of the culture. So you can get very consistent product through the 14 days process. So that's one of the examples.
Now, we have synthetic biology, we have AI. We live in exciting times. There's so much going on. How do you think will these technological advancements revolutionize the design and the optimization of cell factories for industrial use?
I think you mentioned two kind of key technologies or approaches, like is artificial intelligence, and you can add machine learning and synthetic biology. It has so much potential that we're still figuring out how to use those. For instance, artificial intelligence can help you to navigate this enormous amount of data. I think in the last 2-3 years in conference, you talk with the big pharma and they say they're collecting more data in the last year than in the last 20 years. So they are getting to a point that how you can make sense of such amount of data, how you can drive useful information that can be tailored to make your process much better. And I think AI can be very powerful there, like how you can develop algorithms to just intentionally process your data and derive conclusions that can give you some benefit for your process. And for synthetic biology is such an interesting field, and sometimes it's an umbrella that you can put so many things underneath. But I think one of the benefits of synthetic biology, and that something that we're trying to work very hard in here in Manchester, is how you can provide cells with new functionalities. So we're talking about how you can control things, how you can control behavior of cells in a dynamic way. So you can develop genetic circuits that can help the cells behave better in certain period of the culture, and how you can modulate things in a more controllable way. And the other thing that, for instance, we're working on that and other people are working as well, is how you can use the concept of control, or where you have like in a real world manufacturing settings where you have control engineering systems, you have a big process in which you have sensors, and from those sensors you have adapters, and then you have feedback loop of response that you can sense, adapt, and react to the specific stimulus. And you can use synthetic biology to translate those concepts of control engineering systems into the cells. So you can develop cells using synthetic biology to make more robust system in which you can internally sense what is going on, because cells are continuously sensing stress in different ways. But the way that you can sense and adapt and create a response is still lacking on the system. And synthetic biology, it has a lot of potential to develop those systems and make cells that are highly adaptable to different bioprocessing environments.
Wow, that's exciting. I'm hearing that in the future, we will see a lot more convergence of all the different technologies, and we will be able to generate a lot more data, make a lot more sense out of that. So yeah, it would be interesting to have this conversation in a few years down the road and see what actually has happened.
Yeah. What about you? What do you think? I mean, you have a lot of experience not only talking with different companies, you also have worked in a really big company who has been addressing or putting a significant amount of effort to overcome these things. Or just what do you think about it?
I see that definitely the process understanding is improving. If you do it right, if you measure the right things and you set up the right experiments, nowadays you can generate so much data and you can actually make sense out of the data using the right technology. You mentioned hybrid modeling. There's a lot of, obviously, other tools we have at our disposal. So yes, this is one trend I see, to generate a lot of data and then make informed decisions out of the data you are generating. And the other thing I believe is that all these technological advancements will have a multiplying effect. It's not a silo anymore. I think if we look at AI, machine learning, synthetic biology, omics, and a lot more technologies, I think the borders will become much more blurry and we will have a multiplying effect as we're putting these technologies together, and they will enable us to make a much bigger leap faster than we have ever been possible. I'm very excited about the future. We will see a lot of breakthroughs in the future.
Yeah, that sounds very promising.
For our listeners, Mauro, who are not so familiar with cell factories, are there any resources you can recommend?
There are different ways. So scientific literature, but it is a little bit dense. And there are like a few of artificial intelligence-based tools that they can quickly summarize down to these very complex scientific literature. And other things are like really good platforms like Biopharma International, or for instance in the UK there is an association called Bioindustry Association, in which they have bulletins of how these new trends are being implemented in bioprocess industry. I think there are so many that it's very difficult to recommend. One is sometimes is with social media and the approachability of the internet, you can reach out people. And I think most of the people are very willing to just have a quick call or just have a quick conversation about specific topics.
Excellent, Mauro. So we have covered a lot of ground today. What is the most important takeaway from our conversation?
I think there's something that we talked just 2 minutes ago is the future looks very promising. And I guess the merging of the different technology like synthetic biology, artificial intelligence, machine learning, it gives a lot of potential to start thinking about what is the next generation of medicines or the next generation of bioprocess that can be more predictable, making more affordable medicine for patients, and technologies that they are highly robust and highly reliable.
Mauro, where can people connect with you?
To be honest, I'm not really big on social media, but I have my LinkedIn profile, and that has been working well for people who want to reach me out. So yeah, I would say that.
Fantastic. Thank you very much, Mauro, for sharing all your insights. And smart biotech scientists, please reach out to Mauro. His LinkedIn profile, I will leave all the links down in the show notes, also from the University of Manchester. Thank you so much for being on the show today.
Thanks to you for the invitation. It's always interesting to have nice conversation with people in the field, and you have been doing a great work, like putting expert in the public ear. So I really appreciate that.
Hey, smart biotech scientists. Have you enjoyed our conversation with Mauro Torres, who's a chemical engineer and molecular biologist at the University of Manchester? Please let us know what you thought about today's episode by leaving a review on whatever platform you found us on. And do you want to take your process development game to the next level? If so, I have this amazing offer for you. I want to offer you a free 30-minute assessment to help you get started. Just book the call using down in the show notes, and I'm looking forward to seeing you on the call. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.